Welcome to the lecture where all of metabolism comes together. Sugars, fats, and amino acids all converge on a single oxygen-dependent metabolic pathway known as the citric acid cycle, or the Krebs cycle. Even more than glycolysis, the citric acid cycle is a central metabolic pathway where everything comes together. And this one pathway is used by every cell, from bacteria to amoebas to you. We trace the fate of food molecules like sugars and fatty acids through a first set of oxidative reactions to extract energy. For sugars, it was glycolysis that broke the sugars down into 3 carbon molecules called pyruvate. For fatty acids, it was beta oxidation that made the 2 carbon acetyl CoA. Glycolysis generates some ATP, but its main role is to deliver molecules that can be further oxidized to extract more energy. This is where the citric acid cycle comes in, providing a second stage for the efficient production of energy from food. Like glycolysis, the citric acid cycle extracts energy and provides intermediates for other pathways. But where glycolysis and fatty acid oxidation can be compared to a highway or a bike path, the citric acid cycle is like a large traffic roundabout. It's a grand ring road that intersects all the other metabolic pathways. As a roundabout, the citric acid cycle returns to its starting point, and like a roundabout, molecules from other pathways can enter it at various points and leave as needed. Just as in glycolysis, the oxidation of the intermediates of the citric acid cycle is coupled to energy capture, both in phosphate bonds and in molecules of the activated carriers, NADH and FADH2. In this lecture, we will first trace the reactions of the citric acid cycle with an eye to energy extraction. Then we will focus on the ways in which the cycle acts as a hub, connecting intermediates, not only for the breakdown of sugars, fats, and amino acids, but for also converting one amino acid into another, for nucleotide metabolism, and for the urea cycle. Finally, we will explore how the citric acid cycle's connections to diverse metabolic pathways explain some very odd observations about cancer cells, including running the cycle backwards. Now, the citric acid cycle has historically been called the Krebs cycle, named for its discoverer, Sir Hans Krebs, who won the Nobel Prize in Physiology and Medicine in 1953 for his work. It's also called the tricarboxylic acid cycle, or TCA cycle, since the citric acid molecule has 3 carboxyl groups. Let's begin with a look at the 8 reactions of the cycle and the energy-rich molecules produced in them. We arrive at the threshold of the citric acid cycle with pyruvate or acetyl CoA. Most commonly, we have traveled the glycolysis pathway that oxidized sugars to yield pyruvate, or we've chewed off acetyl CoA in several successive oxidations of fatty acids. Now, acetyl CoAs can enter the citric acid cycle directly, but pyruvate made in glycolysis needs an extra step to get converted to acetyl CoA, and then it too can enter. To start the process of entering the pathway, pyruvate needs to pack its bags and move from the cytoplasm, where glycolysis produced it, to the innermost section of mitochondria called the mitochondrial matrix. To get converted to acetyl CoA, pyruvate is first transported into the mitochondria, where an enzyme complex called Pyruvate Dehydrogenase acts on it. Remember, conversion of pyruvate to acetyl CoA is necessary for entry of pyruvate into the cycle. The reason we call pyruvate dehydrogenase a complex is because it is not just one enzyme, but rather several enzymes that catalyze the multiple reactions necessary for converting pyruvate to acetyl CoA. The enzyme complex uses 5 different cofactor molecules that act as helpers for the reactions. These cofactors include lipoic acid, coenzyme A, thiamine pyrophosphate, FAD, and NAD+. The last 4 of these molecules are either vitamins or are derived from them. The main events the enzymes and their cofactors bring about are a molecule of carbon dioxide is released from pyruvate. This is a decarboxylation. Electrons also need to be transferred to electron carriers. This is an oxidation, loss of electrons, and is a 2-step process. First, the electrons are removed, and then they're donated to NAD+ along with a proton to form NADH. Gain of electrons is a reduction. Oxidation-reduction reactions are kind of like accounting. If I transfer money in the bank from one account to another, one account gets debited, the other account gets credited. For every loss, there's a gain, and vice versa. You might wonder what happened to the positively charged NAD+ to make it become the neutrally charged NADH. Well, that's because in cellular oxidations, electrons move in pairs. Thus, NAD+ gains 2 minus charges and 1 positive charge in the H+, to go from NAD+ to the neutrally charged NADH. Almost all cellular oxidations occur this way. Molecules lose electrons in oxidations, and the electron carriers receive the electrons in reductions. As a result of all this, the 3-carbon molecule of pyruvate donates 2 carbons to make Acetyl Coa, which can then enter the citric acid cycle. In addition to sources from sugar oxidation and fatty acid oxidation, acetyl CoA can also be produced by breakdown of amino acids and ketone bodies. Ketone bodies, you may remember, are formed by the breakdown of fat in the liver when carbohydrates are unavailable. Acetyl CoA can also be made by breakdown of the amino acids leucine, isoleucine, and tryptophan. Other amino acids can enter as pyruvate or at later points in the cycle. In short, ketone bodies and amino acids can join the citric acid cycle just like sugars and fatty acids, and this is why these molecules can serve as energy sources when glucose supplies are low. By whichever means it got there, acetyl CoA enters the citric acid cycle and we're off to the races. And though the pathway is a circle, we'll number reactions starting from the entry point for acetyl CoA. So in what we'll call the first step, acetyl CoA transfers its 2-carbon acetyl group to a 4-carbon molecule of Xoloacetate, which comes from the end of the cycle, or what we'll call the end of the cycle. The 4-carbon Xoloacetate and the 2-carbon acetyl group combine to form a 6-carbon molecule called citrate, and it's the name for the citric acid when it's ionized. This reaction to create citrate is very favorable because the breaking of the bond between the acetyl group and coenzyme A releases a lot of energy. This turns out to be important, as we will see when we complete the circle to remake Xoloacetate. In step 2 of the cycle, citrate is rearranged to form its isomer, isocitrate. The enzyme catalyzing the reaction, called aconitase, is the site of action of a poisonous compound known as fluoroacetate. Cells readily convert fluoroacetate into fluorocitrate, and that's a big problem because fluorocitrate is a potent inhibitor of the aconitase enzyme. Inhibiting this enzyme blocks the entire citric acid cycle. And as you can imagine, without the citric acid cycle, cells struggle to harvest sufficient energy from food and may die. Fluoroacetate was used in the US to kill coyotes in the West until the 1970s, when it became apparent that birds feeding on the dead coyotes were themselves succumbing to the poison. Anyway, citrate was rearranged to isocitrate in step 2. Next, in step 3, the isocitrate loses a carbon dioxide and a pair of electrons. That's called oxidative decarboxylation. The loss of a carbon as carbon dioxide converts the 6-carbon isocitrate into a 5-carbon molecule, alpha-ketoglutarate. Meanwhile, the electrons from isocitrate get transferred to NAD+, making NADH again. Remember, oxidation of isocitrate, loss of electrons, causes NAD+ to become reduced to NADH. In step 4, the same thing happens to alpha-ketoglutarate, as another carbon is lost as carbon dioxide. Again, electrons are given away, which combine with NAD+ to form an NADH. The product of the reaction, a 4-carbon compound, links to a CoA forming Succinyl Coa. The enzyme here, alpha-ketoglutarate dehydrogenase, is closely linked to pyruvate dehydrogenase. It uses the same 5 coenzymes and has a similar reaction mechanism. In step 5, the coenzyme A is removed from succinyl CoA to produce succinate. Removal of the CoA from succinyl CoA is also a very energetically favorable reaction, just like we saw in the first reaction when the CoA was released from acetyl CoA. That's because the bond between succinate and coenzyme A is a high energy one. The energy released is used to convert guanosine diphosphate, or GDP, to guanosine triphosphate, GTP. Energy in GTP is equivalent to that in ATP. The remaining reactions in the cycle are focused on recreating the oxaloacetate to complete the roundabout. In the first of these reactions, at step 6, succinate is oxidized to form fumarate by an enzyme called Succinyl Dehydrogenase. As the enzyme name suggests, succinate gives up electrons and protons, which are accepted by FAD to make FADH2. Step 7, the next to last step for completing the cycle, is a simple one. Water is added across the double bond of fumarate to form a molecule called malate. Now, in the 8th and final reaction of the cycle, malate is oxidized to oxaloacetate. And just as in many oxidation reactions, NADH is produced. The reaction, which is catalyzed by malate dehydrogenase, is notable for going backwards under standard conditions. Fortunately, though, the cell is not under standard conditions. You may recall a similar situation with the aldolase reaction in glycolysis. But as you saw in that reaction, removing the product of the reaction enables it to move forward. For the oxidation of malate in step 8, the product gets removed and the reaction gets pulled forward by the citrate synthase reaction, which follows in the next round in step 1. This reaction, as we saw at the start of the cycle, is energetically favorable and removes the product oxaloacetate as it reacts with acetyl CoA to form citrate. With oxaloacetate being quickly removed to make citrate, the production of oxaloacetate can proceed. With that, we've traveled around the cycle and returned to where oxaloacetate reacts with acetyl CoA to make citrate. There are 2 turns of the cycle per glucose molecule being oxidized. That's because each glucose gives rise to 2 pyruvates, which in turn give us 2 acetyl CoAs. Okay, let's look at how much energy we've extracted through the oxidation reactions of the citric acid cycle for these 2 acetyl CoAs. 6 NADHs, 2 FADH2s, and 2 molecules of GTP. And if pyruvate was the source for the acetyl CoAs, we also got 1 NADH for each pyruvate from glucose, for a total of 8 NADHs, 2 FADH2s, and 2 molecules of GTP. Now that we've examined the energy extraction role of the citric acid cycle, let's spend a few minutes talking about its connection to other metabolic pathways. The citric acid cycle links to many pathways, feeding intermediates into some and being fed by others. In its role as a hub for many pathways, portions of the cycle can operate independently, just as you might enter a roundabout and then exit without going all the way around. We'll see a great example of using part of the roundabout in a minute, but let's pause briefly to look at some of the molecules trying to merge on it and exit off of it. Let's start with a molecule that exits. Step 1 of the citric acid cycle, as you learned, combines acetyl CoA with oxaloacetate to make citrate in the matrix of the mitochondrion. As you might guess, lots of citrate gets made when the cycle is fed abundantly with acetyl CoA. When more citrate is made than is needed for immediate energy production, such as when you eat too much and exercise too little, the citric acid cycle gets overloaded. And when this occurs, citrate accumulates and gets transported into the cytoplasm. There, the citrate gets split back into oxaloacetate and acetyl CoA. Now in the cytoplasm, the acetyl CoA is used to construct fatty acids, while oxaloacetate gets recycled back into the mitochondria. And when fatty acids get synthesized, they get bundled together as triacylglycerols, which you know is fat, to be stored for a rainy day or for a New Year's resolution. That's why eating more than you burn gives you what's known in the belly business as a hangover. Acetyl CoA is also the raw material for making cholesterol, and we'll see more about why that's significant in a minute. Citrate that is not split up in the cytoplasm serves as an allosteric effector, binding and activating the most important enzyme in fatty acid synthesis, acetyl CoA carboxylase. So not only is citrate a source of the raw material needed for making fatty acids, it also turns on the enzyme that stimulates the process. The cycle intermediate alpha-ketoglutarate is also an on-ramp and an off-ramp. It has several roles besides being an intermediate in the citric acid cycle. First, and perhaps most importantly, it can grab excess nitrogen and easily be made into the 5-carbon amino acid glutamic acid. Now, from glutamic acid, it's just one step to glutamine. This is just one of at least 10 instances where an intermediate of the citric acid cycle is linked to amino acid synthesis. Many of these reactions can reverse too. Glutamic acid and glutamine can be converted to alpha-ketoglutarate. Thus, glutamic acid and glutamine enter the citric acid cycle via alpha-ketoglutarate. Alpha-ketoglutarate plays a crucial role in nitrogen metabolism. Its ability to accept amine groups allows it to mop up toxic ammonia and to participate in reactions called transaminations. These types of reactions are very important in amino acid metabolism. For example, the amino acid alanine can transfer an amine to alpha-ketoglutarate. In this reaction, loss of an amine converts alanine to pyruvate, while gain of the amine by alpha-ketoglutarate produces glutamate. The pyruvate from alanine can then be converted to acetyl CoA, providing a path for yet another amino acid to enter the citric acid cycle. Our next multifunctional citric acid cycle intermediate is Succinyl Coa at step 4. Succinyl Coa provides an on-ramp for 3 amino acids to enter the cycle, and it's also one of the building blocks used by cells in the synthesis of heme. Heme, as you know, is the flat ring structure found in hemoglobin that is the site of oxygen binding in the body. Fumarate and Xoloacetate also have numerous connections to other pathway molecules. Fumarate is made in both the urea cycle and in the metabolism of purine bases of DNA and RNA. It is also an entry point for 2 more amino acids, phenylalanine and tyrosine. The final molecule I'll talk about here is Xoloacetate, and it's also connected to multiple pathways. It is an intermediate in glucose synthesis. It's also used to easily mop up an excess amine group to become the amino acid aspartate, or 2 amino groups to become asparagine. In the reverse reactions, breakdown of those amino acids can give rise to Xoloacetate, providing entry points for 2 more amino acids. So why do we or why should we care so much about the citric acid cycle? Metabolic connections allow control of the traffic of metabolic intermediates. Having intermediates that interact with other metabolic pathways is not unusual to the citric acid cycle. Glycolysis has that, as do other pathways, but none are as connected to so many pathways as the citric acid cycle. With direct connections to amino acid metabolism, the urea cycle, sugar metabolism, fatty acid metabolism, nucleotide metabolism, and the synthesis of heme, the citric acid cycle is not just the finger on the pulse of the major pathways of the cell, it actually sets the pulse rate. Now let's take a look at how this knowledge is helping to unravel a fascinating mystery about cancer cells that has long puzzled scientists. Get ready for the twists and turns of a biochemical whodunit. The story goes back to the 1920s when Nobel laureate Otto Warburg made a surprising discovery about cancer cells. He observed that they appeared to be using glucose horribly inefficiently, taking in huge amounts and producing similar quantities of lactate. Lactate, as you learned earlier, is a product of fermentation when animal cells don't have enough oxygen. Now, at first glance, there seemed to be an explanation. Cancer cells often do not have a good supply of oxygen because the blood vessel network in tumors is abnormal. Cancer cells, as a result, are often hypoxic or short of oxygen. And that would explain why they make lactate. They're running fermentation since they're out of oxygen. This explanation implied, of course, that if cancer cells could somehow get more oxygen, they would behave differently. But Warburg was in for a big surprise when he provided cancer cells grown in the lab with plenty of oxygen. Under these conditions, when the cells were not dependent on their blood vessels to supply them with oxygen, you might expect them to stop making lactate and start using glucose more efficiently. But strangely enough, that didn't change anything. The cells carried on as if the oxygen wasn't even there, taking in lots of glucose and making lots of lactate. And nobody knew why cells with plenty of oxygen acted like there was none, and to be honest, it's still a bit of a mystery. However, some aspects of the mystery are becoming clearer. For example, we now know that pyruvate dehydrogenase, which you remember catalyzes conversion of pyruvate to acetyl CoA, is inhibited in cancer cells. And that may be a strategy for therapy, as we'll see. Another odd finding about cancer cells also surfaced much later. It turned out that cancer cells, in addition to glucose, also take in large amounts of the amino acid glutamine, far more than they need, for protein synthesis or for using it to make nucleotides. What in the world are they doing with it? Well, this is one of those times where it's great to be a scientist. You shrug your shoulders, say, well, I don't know, and then start applying your knowledge of biochemistry to come up with a few hypotheses to test. Though lactic fermentation is going on in these cancer cells, it doesn't necessarily mean the entire citric acid cycle is non-functional. It's true that pyruvate is not being converted to acetyl CoA to feed the citric acid cycle at the entry point, but remember the analogy of the roundabout. Molecules can enter and leave at multiple points. The cancer cell's heavy use of glutamine could be a clue. Glutamine, let's recall, is readily converted into glutamate, which in turn can be converted to alpha-ketoglutarate. And alpha-ketoglutarate, of course, is an intermediate at step 4 in the citric acid cycle. What might it be doing? Well, this is what scientists at Memorial Sloan Kettering Cancer Center and the University of Pennsylvania wondered. Let's follow their reasoning and findings, first published in 2011, which shed new light on the curious behavior of cancer cells. The most obvious fate for alpha-ketoglutarate would be to continue along the cycle and produce succinyl CoA, succinate, malate, and xoloacetate. But there's a problem with that scenario. Since cancer cells are hypoxic and carrying out fermentation, the NADH concentration is high and NAD+ is low. And 2 reactions of the cycle after alpha-ketoglutarate require NAD+. Without a steady supply of NAD+ in these cancer oxygen-deprived cells, the cycle can't continue as it normally does. In addition, the inhibition of pyruvate dehydrogenase noted earlier blocks an important source of acetyl CoA, so there's another obstacle to the cycle moving forward. So what's left? Well, a less obvious but more intriguing possibility is that the cycle might run backwards from alpha-ketoglutarate. This would reverse the isocitrate dehydrogenase reaction to produce isocitrate from alpha-ketoglutarate, carbon dioxide, and NADH. If going from step 4 back to step 3 were possible, then reversing the even earlier aconitase reaction from step 2 is easy. The outputs of these step reversals would produce citrate and NAD+. And whenever citrate accumulates in the mitochondria, remember, it gets exported out to the cytoplasm where it gets converted to xoloacetate and acetyl CoA. And you remember that in the cytoplasm, acetyl CoA gets made into fatty acids and can also be used to make cholesterol. Both of these compounds are necessary for making membranes, for making new cells. Thus, the reason glutamine may be taken up abundantly is to create acetyl CoA for making molecules needed by rapidly dividing cancer cells. Now, running part of the citric acid cycle backwards seems pretty radical, though. I mean, is that even possible? Well, it turns out that cancer cells do just that. They use alpha-ketoglutarate derived from glutamine to make citrate, which in turn is used to generate other molecules they need to support their growth. In fact, if cancer cells are starved of glutamine, or if the isocitrate dehydrogenase enzyme that catalyzes the backwards reaction was inhibited, the cancer cells were unable to grow and divide. So cancer cells' greediness for glutamine makes sense. What about all that glucose that cancer cells use? Though oxidizing glucose completely to carbon dioxide is more efficient than lactic fermentation, that may not be the main consideration when glucose is abundant. When gas is cheap, for example, people buy bigger, more gas-guzzling cars than they do when gas is expensive. Why should cancer cells be any different? There's another advantage to using lots of glucose, though. When lots of glucose enters a cell, lots of G6P gets made. In the sugar metabolism lecture, we learned that G6P made in glycolysis could take two paths. It could go the glycolysis route and ultimately make pyruvate. Or alternatively, it could enter the pentose phosphate pathway, or PPP. The PPP is interesting because it has a catabolic phase for breaking down G6P to make NADPH. It can also use the G6P breakdown product to make ribose sugars, which of course are needed for making nucleotides for RNA and DNA. Bingo! Cancer cells, which are dividing rapidly, need to make nucleotides to replicate their DNA. Lots of glucose leading to lots of G6P spilling over to the PPP makes good sense for that. So we've accounted for nucleotides and we've accounted for making acetyl CoA. But cell division requires membrane lipids. So the cells would first need to make fatty acids and cholesterol. How do we get from acetyl CoA to fatty acids and cholesterol? We'll talk much more about these pathways in an upcoming lecture, but here's the bottom line. Making them requires ATP and NADPH. In cancer cells, ATP is created by using all the glucose in glycolysis. And here's the clincher. NADPH is one of the prime outputs of the PPP. So let's take a look at how all this ties together. First, rapidly proliferating cells like cancer cells take in lots of glucose and glutamine. Glucose is used for making ATP energy, NADPH for fatty acid and cholesterol synthesis, and ribose, which together to supply vital molecules to rapidly dividing cells. Glutamine turns into alpha-ketoglutarate to feed the citric acid cycle. And last, fermentation conditions raise levels of NADH and slow or stop the forward movement of the citric acid cycle. So reactions run in reverse, starting with alpha-ketoglutarate and resulting in the release of acetyl-CoA in the cytoplasm, where it can make fatty acids and cholesterol. Wow. There may be yet more to this story, but the pieces are starting to fall into place pretty well. The million-dollar question is whether altering what the citric acid cycle does can inhibit the growth of cancer cells. Ongoing investigations are focused on key steps. For example, activating the inactive pyruvate dehydrogenase of cancer cells might force the citric acid cycle to move forwards thanks to acetyl-CoA production. This would also prevent cells from diverting G6P into the PPP, starving them of NADPH, as well as the ribose that they need for making nucleotides. Another approach to stymieing cancer cells is blocking the conversion of glutamine to alpha-ketoglutarate and preventing the synthesis of citrate. It is known that inhibiting isocitrate dehydrogenase prevents cancer cells from dividing because this too blocks the production of citrate from alpha-ketoglutarate. Now, while these studies are still in the early stages, they may turn out to be promising paths to new therapies. Okay, all sorts of food molecules are broken down to be used in the citric acid cycle, where they are oxidized for more energy. In addition, the cycle is also a hub for intermediates that can feed or draw from every other major metabolic pathway. This one roundabout is central to the business of oxidative energy extraction and is at the heart of all of metabolism. That's why reversing this one path goes so far towards explaining the bizarre behavior of cancer cells. The NADHs and FADH2s made in the citric acid cycle are ready to pass on their electrons to the electron transport chain, which will be the final leg of the energy extraction story. The electron transport chain is where the vast majority of the energy captured by oxidative metabolism in the citric acid cycle is finally converted to ATP. The citric acid cycle is a source of energy. It gets electrons moving while reducing NAD. It starts with citric acid, turning to a conittate, which becomes an isocitrate on the way to glutarate. The loss of one more carbon gives succinyl Coa, and then succinic acid when the Coa goes away. A further oxidation gives one transfumarate, which gains a water on the next step to make malate. One simple oxidation makes OAAUC, which combined with ACOA returns acyclically.